I'm going to disappear here for a second. And I'm going to take you on over to the schedule. Every chat session, I would like to just kind of do a quick reminder for the schedule for you folks. So there's no questions. As of right now, you should have completed all of these items. The Art and Science, School of Art and Science student information sheet or acknowledgement form, okay? One page, real simple, like your name and what you're studying, blah, blah, blah. Required information quiz, your discussion board, part A. Okay, that is basically you introducing yourself to the class and just telling us a little bit about yourself. Real quick, that's probably about 6 to 10 sentences. And then Smart Book assignments 1 and 2 for chapters 1 and 2. Just to remind everybody, the Smart Book is a graded assignment upon completion. So we call it an all or nothing assignment, meaning if you finish it, you'll get full credit. It's really easy. If you don't finish it, you will not get any credit. Doesn't matter if you've answered 35 out of 40 questions, 20 out of 25 questions, you have to complete the whole thing. It's a very useful tool in learning, trust me. Okay, I've been doing this for quite some time and I've had several students really enjoy it. They might not enjoy it at the time, but they see the benefits afterwards. So trust me, the Smart Book will be helpful to you. This is all geared to help you become successful in this course. This coming up Sunday, you will have 3 assignments. Smart Book Chapter 4, your first quiz, Connect quiz for Chapter 4. There's no Connect quiz for Chapter 1 or 2, none. This is your first quiz. All right, that's the Chapter 4 Connect quiz. And then the second part of the discussion board, part B, introduce yourself, where you go and you have to respond to at least 2 of your classmates' introduce yourself to the class discussion board posts. Okay, so you're going to find 2 of your classmates, answer with a couple of sentences, please, so we can see that there's some dialogue and some interaction going on. Okay, that being said, let's get back into review. Whoops, that's wrong. Give me 1 second here. Okay, so I'm hoping by now that you folks have had a chance to kind of go through some of the material, read these announcements that I send out on Sunday or Monday nights. They will tell you what you should be doing during the week. I will send out a little bit more of a lengthier announcement in the middle of the week, right? Kind of honing in on what you should be covering. So this is to help you so you don't fall behind and so you kind of know where you should be when you're learning this material here. So today we're going to talk about Chapter 4, and Chapter 4 is a crucial chapter because it discusses the cell, the smallest organic or living functional unit in our body. And a lot of the physiology is dependent on the actual functionality of these cells. So basically, when we're talking about these physiological processes like secretion, absorption, filtration, that is dependent on the cells doing what they are supposed to be doing. Okay, so this is a great test question right here. Cells are known as the functional units of the body. I love throwing that onto tests, so you should definitely know that. And so as you know, with this course, the study of anatomy and physiology, they're integrated. You mix them together. So if you understand the cell structure, you will definitely understand the function of the cell easier. So we'll get into some of that when we get into tissues, but we're going to talk about the real basic components of our cells. Some of you may have never, ever learned anything about cells, and that's okay. That's okay. We're gonna work you through it. So let's start off with some of the common features of the cell. And there's 3 parts to the cell. Okay, you got your plasma membrane, you've got your nucleus, and the cytoplasm. So a couple things. The plasma membrane is the outer covering to our cell. All right, the nucleus is this big ball that's inside the cell. It's referred to as the control center. And it's sometimes in the middle, sometimes it's to one side, at the bottom of the cell, whatever. And then in the space between the nucleus and the plasma membrane, that's the cytoplasm. So the cytoplasm, we'll talk about the components of the cytoplasm, all right? But that's going to be kind of everything in between the nucleus and the plasma membrane, inside the cell. So you're going to really learn a lot about the different functions of these structures here. But the plasma membrane, you know, when I first took cellular biology in high school, didn't really appreciate, you know, its functions, its purpose. You're going to learn the plasma membrane plays a huge role. All right, but first, let's learn a little bit about it, okay? It's the outer coating, and it's very important that you understand this term here, limiting, okay? It's going to limit the movement of substances into the cell and out, and moving from outside the cell into the cell and inside the cell, outside the cell. Basically means it's going to separate the internal cellular environment from the external cellular environment. And it's important, okay? So we're going to, throughout this course, we're going to discuss its role and how it is going to do certain biological functions here. On some of our plasma membranes, you'll notice that the surface has these extensions, these modified extensions, and there's three types: the cilia, flagellum, and microvilli. The first two that are listed here, the cilia and the flagellum, have to do with movement. The microvilli does not have to do with movement, but it actually increases the surface area of the cell, and it helps to increase absorption. So we'll get into that later on. So when we're talking about the cilia and the flagellum, and I said movement for those two structures, okay? One of those structures will actually propel and move the cell, and the other one, the cell is stationary and it moves things past the cell. So the flagellum actually is like a tail-like appendage, and it kind of moves and it propels the cell. There's only one cell in the human body that has a flagellum, and that's the male sperm. It's the only cell. The cilia are these hair-like appendages, and they kind of whip back and forth. And what they'll do is they'll move things past the cell. For example, mucus in our respiratory passageways. Okay, you're always producing mucus, right? Because mucus actually serves as a protective structure, all right? And so we don't want to building up in any particular surface, especially if you have a cold and whatnot. And so the cilia moves the mucus away from the lungs towards your trachea. So that's one example. Okay, the nucleus. So when you're looking at a cell, this is the largest structure that you can see inside the cell. And the nucleus is similar to the actual cell itself. It'll often mimic the shape of the cell, all right? But also, it too has its own outer coating. We call that the nuclear envelope, and that covers the nucleus. But it's important because inside our nucleus is where we keep our DNA, our genetic blueprints, what makes us us. So when it's time for us to make proteins for whatever the function of our cell is going to be, or if we need to have the cell divide, all right, well, all that material, okay, down to the slightest detail, color of your eyes, color of your skin, how tall you are, all right, all of that is encoded into our genetic material. And so that's stored in each individual cell. So also inside the nucleus, we have what we call a nucleoplasm. It's similar, not the same, but it's similar to the cytoplasm. It's like a goo-like material that sits inside the nucleus. And then the last part of our cell is the cytoplasm. And this is going to contain all the structures in between the nucleus and the plasma membrane. And so there's three parts to it. The cytosol, which is actually the liquidy portion of the cytoplasm. The organelles, which we'll talk about. Okay, these are small little structures that help with certain functions of the cell. And then everything else that we just throw into this category, which we call inclusions. And we'll get into that. Okay, so let's talk about our cytoplasm. We'll start with the cytoplasm. Remember where the cytoplasm is. It's between the plasma membrane and the nucleus. And so there's three components: the cytosol, the organelles, and the inclusions. Another name, and you need to know this, for cytosol is called inside, intracellular fluid. So if you're reading a sentence and you came across this term that said extracellular fluid, then you would say, okay, I already know what the intracellular fluid is. That's the cytosol. Extracellular fluid is all the fluid outside the cell. So you need to know those terms: intracellular fluid inside the cell, extracellular fluid outside the cell. So it's going to be goo, all right, like a viscous fluid. But of course, like with most things in the body, when we have fluid-filled compartments, it has a high water content. And so certain structures, if you read chapter 2, you're going to know that certain solutes, certain things can dissolve in water. If they're able to dissolve in water, we call them hydrophilic, water-loving, and usually they are polar, and they are, or they can be charged like ions. So certain polar macromolecules, if you're wondering what a macromolecule is, a macromolecule is a protein, lipid, okay, carbohydrate. Those are macromolecules. So in the cytosol, you'll see that some of these macromolecules have dissolved into the cytosol, and certain ions like sodium, potassium, calcium, and those will also be dissolved. And so for that to be dissolved into the cytosol, because it has a high water content, that means it has to be water soluble. So that means it has to be polar, right? That means that something has a positive and a negative charge on it, or like an ion. Non-polar substances are water insoluble. They will not dissolve, like oil. When you're boiling water for pasta and you pour some olive oil in, okay, you notice that that olive oil does not disappear or dissolve into the water. It kind of floats there on the top. It's water insoluble. It's hydrophobic, water fearing. Important that you know that. Okay, the next structure inside of our cytoplasm are going to be the organelles, little organs. Now, these organelles have various shapes and they have various functions, different jobs to do. And they're quite complex, depending, certain ones are a little bit more simple than others, but some are quite complex. And you think, well, wow, the cell is pretty small and simple. How is it that it's so complex? I'm telling you, this stuff it's fascinating. All right, we're still learning about some of these, the functions, all the functions of some of these organelles. But we break these organelles down into two categories. Membrane-bound organelles, which means that these organelles have a covering or a membrane around them, and non-membrane-bound organelles. So we're going to discuss some of that. So let's start off, going into a little bit more detail about our membrane-bound and our non-membrane-bound organelles. All right, these membrane-bound organelles that have this covering, it's going to separate the membrane-bound organelle from the cytosol. And usually that's because whatever function that these organelles are engaging in, they need a little bit of privacy, all right? And they cannot have whatever they're making or doing, whatever their function is, all right, could be potentially harmful to the cell. So we want to kind of secure them and we want to partition them off. So you'll see that some of these membrane-bound organelles include the endoplasmic reticulum. We'll talk about all this stuff, the Golgi apparatus, lysosomes, peroxisomes, and mitochondria. Now, our non-membrane-bound organelles, you can pretty much figure those out. Those are not going to have a membrane around them. They are made up of protein. So ribosomes, the cytoskeleton, centrosome, and then the proteosomes. These are made of protein, and we'll talk more about this. And then finally, the last part of our cytoplasm are the inclusions. The important thing that you understand about it is that the inclusions are only going to be in the cytosol for a short period of time. They're temporary, okay? So that means that these inclusions, these molecules, these structures will come and go, in and out all the time. For example, glycogen. Glycogen is the stored form of glucose. And so there's two organs in your body that store glucose, your liver and your skeletal muscles. You need to know that. Okay. So the cells of the liver and of the skeletal muscle will take the glucose and store it in the form of glycogen, which is a polysaccharide. And basically what that is it just takes glucose molecules and it starts to make a long chain or string of them. And then when it's time to use that glucose for energy, it just cuts off, all right, those individual glucose monomers, right, those individual glucose molecules, and it uses them for energy. So it comes and goes. So that's what we're seeing here when we're talking about the inclusions. Here's a beautiful picture of our cell. All right, so you can see here, here's our nucleus. Here's the cytoplasm, and then everything in between, excuse me, the plasma membrane. And then everything in between is the cytoplasm. And you can see all these different structures inside the cytoplasm. This kind of bluish, this is the cytosol, the liquid here. Right? And then you have all these different organelles all over the place. So there's a lot going on inside the cell. Our plasma membrane, here are the extensions here. All right, this very long extension is the flagellum. It's a long tail-like structure that will propel and move the cell. Human sperm, only cell that has that. Then you've got something that's shorter than the flagellum, but longer than the microvilli here. These are the cilia. And while the cell stays stationary, the cilia move back and forth and it'll move things past the cell. And then you've got the microvilli here. And you can see basically the plasma membrane is just poking out. Okay, but you can see that, look at all of this plasma membrane that occupies such a little amount of space. And it's designed to increase the surface area of the cell for certain functions like absorption. So we can get more absorbed into our cell. All right, so let's talk about some of the general functions for our cell. These are very important. And there's usually a test question or two based on the cell's general functions. And I've seen one, this first one here, I've seen it asked before on a test. All right, so some of the general functions for cells are going to be to maintain the integrity and the shape of the cell. And that's important. As cells become damaged or weaken, they can start to lose their shape, and the plasma membrane may become damaged and then expose the internal environment to the external environment, and then that's when problems start to occur. Right? There are certain cells, immune cells, that like to wander around, and they find a cell that's infected with something, then they basically start to punch holes in the infected cell. And when that happens, that infected cell will then start to swell and die. Parts of it will leak out. So it's very important that the plasma membrane maintains its integrity. And so therefore it'll maintain, all right, what's going on inside the internal environment. Another cell function is to obtain nutrients, right? Just like us, we have to obtain nutrients to actually satisfy our metabolism needs. So the same thing happens to the cell. It's got to obtain nutrients, and then it's going to use those nutrients to make things. Either things for energy, so it can actually undergo certain types of functions. If it has to make certain hormones, let's say, or if it needs to make certain proteins, okay? It needs those nutrients to form those building blocks there. And then, of course, just like us, if you eat something, you're eventually going to have to get rid of it and go to the bathroom. Well, these cells do the same thing. They're going to get rid of any wastes, all right, as a result of their metabolic processes. And it wants to avoid the accumulation of those wastes inside, because if it starts to build up, they can reach toxic levels, and then that could be harmful to the cell and cause damage. Then we'll find out that certain cells can divide and others cannot. Those that are capable of cell division, all right, will undergo cell division because we might have to replace damaged cells, or if you're growing, we need to create more cells. And so this is all part of the normal processes of our cells here. Now, some cells do not divide. We call those amitotic cells. Perfect example are going to be your nerve cells or neuron, right? They, after a certain point, you know, after the fetal development, right, they don't divide anymore, except there's always exceptions to the rule in extreme cases. And we'll address some of those later on. But for the most part, certain cells don't divide. All right, so let's talk about some of our organelles inside the cell. And we're going to start off with our membrane-bound organelles, and I want to talk about the endoplasmic reticulum. So if you look here at our picture, the endoplasmic reticulum is going to be this tubule network that sits usually in close proximity to the nucleus. So the nucleus has the DNA, and so sometimes if that cell needs to make proteins, for example, all right, we need to get that information off the DNA, and then we're going to transcribe it onto RNA. You've probably been hearing a lot about mRNA, okay, with the new vaccines that are out there. And basically, that's what mRNA does, and that's what that vaccine is designed to do. It's designed to produce proteins inside of your cell, all right? The spike protein, for example, with COVID. So your own cells are making the spike protein, and then your immune cells identify that spike protein as a foreign substance. And if it's a foreign substance, then it wants to destroy it. And so that's how we mount our immune response. And that's how you get immunity to the COVID. Okay, so that's what we're seeing here in our endoplasmic reticulum, right? The endoplasmic reticulum is going to help us make proteins, and those proteins will be secreted into 3 places, all right? When I mean secreted, it's going to actually take those proteins that they make and do one of 3 things with it. It's either going to put those proteins into the plasma membrane, okay? Or it's going to take it and package it up and send it off to the Golgi Apparatus. We'll talk about that in a moment. The Golgi Apparatus is like a warehouse in which it'll modify certain proteins and package them up and send them to be secreted outside the cell. And finally, the third place that the proteins that are made here in our endoplasmic reticulum, the proteins will go into these structures, which we'll talk about here in a few moments, called lysosomes. And it'll make these enzymes for these lysosomes. They're basically digestive enzymes. And so they're important. Like, for example, if we're trying to make thyroxine, all right, during the whole process of making our thyroid hormone, we have to have the lysosomes in those thyroid cells interact with, all right, our thyroxine to do a couple chemical reactions to make it ready to be exported out to the body. So those are the 3 places in which the proteins that are made here in the ribosomes, or excuse me, not the ribosomes, in the endoplasmic reticulum are going to wind up. So some of the functions that you can see here for the endoplasmic reticulum is going to be synthesis, all right? We're also going to see, all right, when we're making, well, first of all, let me backtrack here a little bit. If you look at the picture here, you can see our rough endoplasmic reticulum and a smooth endoplasmic reticulum. The difference is one has ribosomes, these little balls here. Ribosomes are proteins that help to make actual protein. And then the smooth endoplasmic reticulum does not have any ribosomes here. Let me go into a little bit more detail. Did I skip that? Hold on one second. Yep. All right, let me go back. All right, so with the rough endoplasmic reticulum, we're going to make proteins that are either going to go into the plasma membrane, or they're going to be enzymes for the lysosomes, or they're going to head over there to the Golgi apparatus and be packaged up and modified for export out of the cell. All right, so keep that in mind. We're also going to see, all right, this is the area where we're going to start to make peroxisomes. We'll get into more of that in a little bit. You have all these little structures or organelles that are highly important in the making of certain substances and the actual degradation or degrading of certain substances. And we'll see here in the rough endoplasmic reticulum the creation of what we call transport vesicles. Think of those as like a railroad car, right? It's going to transport a substance from point A to point B. Now over here, the smooth endoplasmic reticulum, this is where we're going to see, all right, the creation or the synthesis of some of our lipid molecules, all right, cholesterol or some of our steroid hormones will occur here in the smooth endoplasmic reticulum. We'll also see the creation of glycogen. So the smooth endoplasmic reticulum, you'll see the formation of the glycogen molecule, which is the stored form of glucose. We'll also see quite a bit of detoxification of drugs and alcohol and some poisons. Then, of course, we'll also see more vesicle formation there. So you heard me talking about the Golgi apparatus. Here it is. This is our cell warehouse here. And the Golgi apparatus, if you look down here in this picture, you've got a nice fat bottom portion and then kind of a skinnier top portion here. So the part that's closest to the nucleus, we call that the cis face, CIS face. And then the part that's furthest away, that's closest to the plasma membrane, that's called the trans face. So one's like the entrance and the other's like the exit. So what will happen is the rough endoplasmic reticulum will make a protein, it'll package it up, and it puts it into these transport vesicles. And they'll ship it off here to the cis face of the Golgi apparatus. And so it fuses with the cis face, the protein contents get emptied into it, and it undergoes, all right, at different parts of the Golgi apparatus, these different chemical reactions. And so they'll modify, add some things to the protein, take some things away, and it'll do a lot of different types of processes to this protein here. And then eventually, when it gets up here to the trans phase, it will repackage it into what we call a secretory vesicle. Okay. And these secretory vesicles get released from the trans phase here. And what will happen is, all right, they'll have one of two things that they can do. All right, these secretory vesicles can actually merge with the plasma membrane and take those proteins or the contents there and actually include them into the plasma membrane, or that secretory vesicle can again merge with the plasma membrane and its contents will get shot out and excreted into the extracellular space, or also known as the interstitial fluid. So the Golgi apparatus does quite a bit, right? It's going to modify these proteins here, and then it'll actually package them up and get them ready, right, for either incorporation into the plasma membrane or secretion from the cell itself. And one more thing you notice down here, we can see that some of these secretions here get packaged up into another transport vesicle. And what will happen is this transport vesicle will fuse with a lysosome, and it gives the lysosome some of those digestive enzymes that the lysosome needs to do its job. So there's a lot going on, but you have to understand the rough endoplasmic reticulum is going to supply that material to the Golgi apparatus. Okay, the rough endoplasmic reticulum, not the smooth. All right, so let's talk about some of these little bubbles that are floating around inside the cytoplasm, the lysosomes. These guys are cool. And it's really important that you understand, all right, what they do. And it's real simple. Basically, what they will do is they will contain the digestive enzymes that they just received from the Golgi apparatus. And what will happen is they just float around the cell, okay? And they will break down molecules that they fuse with, all right, that was maybe hanging around inside the cell that it didn't need. So what will happen is certain substances may enter into the cell. The lysosome will then fuse with that, and it will digest whatever those contents were. So if we have any damaged organelles, like say one of our Golgi Apparatus break down, well, the lysosome will help and get rid of it, okay? Or if you have, you know, just some proteins that were lying around that were going to be used. It's like a building that started to be built but was never finished, all right? Some of the cellular components there, okay, that maybe had been started to be built but never finished, or something starts to break down, no problem. This guy comes along, all right, and will start to digest, all right, these unused or these half-built, all right, cellular components. So they just go around and kind of clean things up. But in some cases, when the cell takes something in from the outside, these lysosomes will fuse, like I was saying with the thyroid hormone, right? It's going to fuse with the vesicle that contains that thyroxine, the inactive thyroxine, and it'll fuse with that, and it'll undergo some chemical reactions to actually finish up the process of making thyroid hormone. Another type of membrane-bound organelle, and I should bring this to your attention too, okay? Think about it. These organelles have a membrane coating because when they contain these digestive enzymes, if these digestive enzymes were just allowed to run freely through the inside of the cell, they'll just start to tear things apart that should not be torn apart or destroyed. So that's why we need the membrane around this organelle to keep it contained so it doesn't escape. All right, so the peroxisomes, another membrane-bound, all right, structure. They're going to be a little bit smaller than our lysosomes here. All right, so you'll see these guys coming off of the rough endoplasmic reticulum, and basically they're going to contain oxidative enzymes. Now, what are those? I'm going to tell you something. Oxidative enzymes are an enzyme that are very powerful in their ability to break down molecules. I'm talking about very, very powerful. Okay, it's like the Uber, like if you want to get a cleaning solution for your house, this would be like the Uber cleaning solution. All right, so again, they'll have a role in some of the chemical digestion similar to lysosomes. Okay, they're going to play a big role in beta oxidation. All right, what is that? That's when we start to break down fatty acids. And so that process helps us, all right, to get individual fatty acids for energy. We'll talk more about that in chapter 27 and 211 when we're talking about metabolism. Okay, but for right now, just think of beta oxidation is that is going to be an energy process for the cell. And so they start to help with breaking down these fatty acid chains. Lipid synthesis, all right, these peroxisomes will help with making, all right, specific types of lipids. For example, bile salts. It's a huge, important lipid because they help in the emulsification of fat. So bile salts help with breaking down fats. So this lipid synthesis process here helps us in trying to make these bile salts. And then they'll also offer in detoxification. All right, that's hydrogen peroxide. For those of you that are familiar with hydrogen peroxide, if you have a cut, you put hydrogen peroxide on it, it helps. And it's because that hydrogen peroxide is highly reactive, okay? And it will help with sterilizing cuts or wounds and whatnot. Or if you want to dye your eyebrows, you can use that too. Some people use it to dye their hair, all right? So we'll see in these peroxisomes, you'll have that hydrogen peroxide, right, and it will help with breaking down of certain molecules. So peroxisomes, their main function is digestion, so breaking some things down, while at the same time, they help with making or the synthesis of other types of macromolecules, pretty much the lipids. One of my favorites, the mitochondria. Know this term, powerhouse. Mitochondria are the powerhouse of the cell. This is where we make a lot of energy. All right, and that's pretty much what it does. Its job, and this is important, you'll need to know this, is to produce ATP, which is the energy currency of the cell. All right, specifically, it makes ATP through the process of aerobic cellular respiration. The first word there is crucial. Okay, aerobic means with oxygen. So if we have oxygen present, we can make ATP. And if that oxygen allows us the ability to make a lot of ATP. So these mitochondria are scattered all throughout the cell. And so that's their job. What is their job? Make ATP. And it'll make a lot of ATP with the presence of oxygen. So it'll undergo aerobic cellular respiration. Ribosomes. These guys here, real simple. Look down here. Protein synthesis, done. That's what they do. Okay, the ribosomes, there's two types. We have bound ribosomes, and those will be the ones, I'm going to go back here real quick. Those will be the ones that you see here. You can see these little bumps on the rough endoplasmic reticulum. Those are bound ribosomes. They sit right on the endoplasmic reticulum. And then you'll have free ribosomes. They've escaped the rough endoplasmic reticulum. And so you'll see them out here, just floating around. There's a difference between bound ribosomes and free ribosomes. Bound ribosomes are going to do one of three things. I talked to you about this before. Okay, they're going to make proteins that are either going to go into the plasma membrane, be exported from the cell, or they're going to make the enzymes, all right, that are going to go inside the lysosomes. Those are the three things that bound ribosomes make. If you're a free ribosome, then you're going to make proteins that are going to be used inside the cell, within the cell. Be able to make that distinction. But so ribosomes are proteins that make other proteins. That's their job. Okay, so they're going to get what's called messenger RNA, and this messenger RNA is going to come along. We're not going to talk about the process of transcription and protein synthesis, okay? But it's going to come along, and this messenger RNA is going to tell these ribosomes what type of protein they're going to make, and they'll start to make it. Okay, now let's move away, okay, from the membrane-bound organelles and get into our non-membrane bound organelles. And this is the centrosome. Now, if you've ever taken cellular biology, I did in high school, cellular biology, the centrosome, the only time we learned anything about the centrosome was that they were present during cell division. And they would make the mitotic spindle. And basically the mitotic spindle is the centrosomes. They would appear when it was time for the cell to divide, and they would go to opposite ends of the cell. So here's your cell, and one centrosome would go over here, the other centrosome would go over here. And what they would do is they would shoot out these little spindles towards the center of the cell. And these spindles would basically pull at the center of the cell and split the cell down the middle, making two new cells. And that's what the mitotic spindle does. And that's what the centrosomes do. So they also have another function, okay? They are going to create these microtubules. And we'll talk about the cytoskeleton here. There's different, a cytoskeleton is the cell's skeleton, its structure and support system. And so these centrosomes will help to make these microtubules, okay, to help, all right, with the support system of the cell itself. But normally, when I first learned about the centrosomes, it was during cell division. All right, you got another structure here called a proteosome. Now, a proteosome is made up completely of proteins. But I love what the proteosomes do. Proteosomes are awesome because their job is for quality assurance. So here's what's going to happen. These proteosomes are floating around in the cytoplasm, and they'll come across a protein here that was tagged with this protein called ubiquitin. And basically ubiquitin, if you get marked with ubiquitin or tagged with it, that's the mark of death. You're gonna die, okay, if you're a protein. So say a protein chain was being made and it never got finished, something happened, and you're just floating around in the cytoplasm, just minding your own business. Well, ubiquitin comes along and it gloms onto you, all right, and it says, nope, this protein's gotta go. And so these proteosomes are floating around, they see the ubiquitin, and basically they're gonna pull that protein into the actual organelle, and it just chops it up. And you know what the building blocks for protein are: amino acids. So it's going to chop up these proteins either into the individual amino acids, all right, or into what we call peptide fragments, which are going to be a couple amino acids stuck together. So hopefully it'll be able to chop off all the individual amino acids, but sometimes it doesn't. So you're left with groups of 2 or 3 amino acids stuck together in peptides, okay, in these peptide fragments. And so that's what they do. They go around, if they find some proteins that were damaged, or again, we won't really discuss it in this course, but when proteins are made, they can go through some modification changes. They get folded into different configurations. Well, if it was folded incorrectly, like if you're doing, what is it called, origami, when they fold all those the paper to make little shapes of like birds or whatever, or animals, okay? Well, this one got folded incorrectly, it becomes non-functional, okay? And so it gets marked with ubiquitin and it gets the mark of death. All right, so you heard me mention the cytoskeleton, and just real quick on the cytoskeleton, you wanna know, when we're discussing the cytoskeleton, obviously, you know what your skeleton is used for. So the cytoskeleton is not much different, okay? It definitely plays a role in the support system of the inside of the cell, helps to give the cell its shape. It also plays a role in organization. It helps to organize these organelles that are just kind of floating around in the cytoplasm. When it's time for the cell to divide, we talked about that when the centrosomes, all right, the cytoskeleton, all right, is utilized in that the centrosomes will create the mitotic spindle. And that mitotic spindle plays a role in the division of the cell. And then for certain structures, like those transport vesicles, for example, when they're moving around inside the cell from the Golgi apparatus to, say, the plasma membrane, well, we utilize the cytoskeleton to help to move those structures. So there's 3 types. There's the microfilaments, the intermediate filaments, and the microtubules. The microtubules are the smallest. The microfilaments are the largest. The cilia, for the modifications to the external surface, I already talked about this, the cilia, the flagella, and the microvilli. Okay, the first 2, think movements. Hair-like structures for the cilia, and they're going to move things past the cell. Flagella is going to propel the cell. The entire cell is moved because of the flagella. And then the microvilli, again, no movement is related to this modification, but it does increase the surface area of the cell, which is very helpful if we're undergoing digestion and absorption. All right. That's for the cytoplasm. 1 of 3 structures out of the way. Let's talk about the plasma membrane. The plasma membrane is the outermost covering to the cell. It is a limiting barrier, which means it controls the movements of things in and out of the cell. Interesting fact, the plasma membrane, and this is important for you to understand, it's a fluid mixture. What does that mean? Well, what it means is that I like to use it, for example, think of a big pile of trash floating on the ocean. Now, I wish I could remember the name of the there was supposed to be some big pile of trash out in the Pacific Ocean somewhere, where all this garbage had just kind of accumulated together. And they called it like Garbage Island, or I can't remember. There was a certain name for it. But anyways, all that garbage floats up and down on the surface of the ocean. That's what the plasma membrane is. It's going to look somewhat like that. It'll be kind of a fluid-like movement to it. It has equal parts of lipids and proteins by weight, not by number, by weight. So roughly 50-50, all right, weight of lipids to proteins. All right, an important fact, and you need to know this. Remember what I said, the plasma membrane allows certain substances to move in and out of the cell. Well, some things don't have to wait for the plasma membrane to let them in or let them out. And they have to have these qualities. They have to be very small, which makes sense. You get small little structures that can move in between things, all right, in the plasma membrane, and they have to be non-polar, okay, which means they have to be hydrophobic, water-fearing, okay, or water insoluble. So they're either small or non-polar, and they can move into or out of the cell without any help at all. So when we're looking at the lipid part of our plasma membrane, there's a couple different types. Phospholipids make up the largest majority of lipids in the plasma membrane. Then we have our cholesterol, which helps in the temperature resistance. And what I mean by that is that cholesterol makes the plasma membrane more hardy or able to withstand changes in temperatures to that plasma membrane. And then we have glycolipids, which are basically just fats with a carbohydrate group attached to it. So here you can see a nice picture of the plasma membrane. You notice here, these little purple balls here with the little tails hanging down, those are the phospholipids. And so if you look at the picture, quite a few phospholipids. And then what we've done is we've got some glycolipids here. Here's that carbohydrate group here. And then you can also see the cholesterol, which is these guys right here, these little yellow globby looking structures. And then we'll talk about the proteins here. These are these big blue things that are floating in our plasma membrane. All right, so remember, the other half of the weight of the plasma membrane is made up of our membrane proteins. And so they're going to float in the plasma membrane, what we call the fluid bilayer. And we call it the fluid bilayer because if you notice here, you've got one phospholipid here, all these little purple balls with the little tails hanging off. And then on the other side, you've got another layer of phospholipids here. So they're basically two layers. And the purple are the heads, and the tails here are the yellow little stringy-like structures. So you have two layers, and the tails face each other, and the heads will face away from each other. This head here is facing the outside of the cell. This head over here is facing the inside of the cell. Whereas the tails are going to be in between in this little region here. All right, so when we talk about our plasma proteins, these plasma proteins are going to do a lot of work for our plasma membrane. They're going to be performing most of the functions. And if you recall, all the functions for proteins when you were reading chapter 2, okay, and if you haven't had a chance, I would suggest you can go to my YouTube page, Dr. Kaz, and I do have some videos on chapter 2. All right, we're not covering it in our chat session here, but you can go to my page there, and if you want to go over there should be a review. Go under chat sessions. I have some playlists there. You can go under chat sessions, or you can go under review sessions and look for chapter 2. I don't have reviews for all the chapters. I do for most. Okay. So there's two types of plasma membranes, integral and peripheral. Integral go across the whole plasma membrane right here. This is an integral protein. A peripheral protein just sits on either the inside of the cell or on the outside of the cell. Here you can see a nice peripheral protein on the outside of the cell. Here's a nice peripheral protein on the inside of the cell. And then you've got your integral protein that spans the entire width of the plasma membrane. So some of the categories here for protein functions, all right, you can see the first one here are transport proteins. So these proteins, real simple, they're going to regulate what's coming into the cell and what's going out of the cell. And so we have a couple different examples: channels, carriers, pumps, symporters, antiporters. We're going to talk about all these. We're going to spend a lot of time on channels and pumps this semester, all right? In 211, you address carriers, symporters, and antiporters in a little bit more detail. But we're going to focus a lot on channels and pumps. Okay, but basically what they do is they regulate the movement of things in and out of the cell. Then we have our cell surface receptors. Okay, the cell surface receptors are going to be found on the external, I shouldn't say just only the external portion, but for the majority. And a lot of times these cell surface receptors allow, all right, the cell to bind onto other chemicals or molecules, and we call those ligands. So we're going to be talking about in chapter 12 neurotransmitters, right? And so we're going to learn how a neuron can stimulate a muscle cell to contract. And that has to do with the cell surface receptors there, because the neuron is going to release these chemicals, and then they're going to go and bind onto the muscle cell and stimulate that muscle cell to contract. And it utilizes the cell surface receptors there. Identity markers are important, especially when we're talking about immune system function, because the identity markers tell our body which cells belong to our body. And if they are not the correct identity marker, then our immune system identifies that cell as a non-self cell or a foreign cell, and it's going to kill it. So it helps our immune system to tell the difference between healthy cells, all right, and unhealthy cells. And then therefore, if it's unhealthy, we destroy it. All right, we'll also discuss the other functions here for our plasma membranes. Enzymes. Enzymes are going to be the catalyst for chemical reactions. So what is a catalyst? A lighter. How do you start a fire? You use a lighter. You light some paper in the fire, and there you have your fire. That's what the enzymes are going to be. They're going to help to start these chemical reactions, and they can even, in some cases, speed up these chemical reactions. So they play a very important role. All right, they offer anchoring sites, so that cytoskeleton that we were talking about a couple slides ago. So if we want to maintain the shape and structure and support of our cell, sometimes we have to have these anchoring sites on the inside of the cell to attach the cytoskeleton to, so we can have our proper support and shape of our cell. And then if you, when we get into epithelial, into different types of tissue, our epithelial cells are closely packed cells. And so these cells attach themselves to one another. And so these proteins help with that. They're going to create these attachment points so we can stick these cells close together. And so that will help with preventing things from moving in between the cells, because guess what? You don't want viruses, you don't want bacteria trying to slide in between the cells. That's bad, okay? Because we don't want that to get to the internal tissue. That'd be bad, like if viruses and bacteria could just simply squeeze in between the skin cells that we have. So we have these cell-to-cell adhesions, and proteins will play a huge role. All right, so you can see here are just pictures of all those different types of functions of plasma proteins that I was describing to you. Here's a transport protein, a channel, something on the inside of the cell, on the outside of the cell, enters the inside. All right, these carrier proteins are actually going to move, for example, glucose from outside the cell to inside the cell. Pumps, same thing, will move stuff in and out of cells. Cell surface receptors, here's that chemical ligand that when it binds to the receptor might initiate some chemical reactions or some physiological responses. Identity marker, this is how our cells are able to tell what is going to be a cell that should be there, what we call a self cell, SELF, or a non-self, foreign cell. Okay, so here's just a couple examples here. All right, so the next concept that I want to discuss is going to be what we call membrane transport. Membrane transport, remember what I said, the plasma membrane is a self-limiting barrier, meaning that it's going to control what goes into the cell and what comes out of the cell. And so to do that, it has to regulate things that are moving in and out of the cell. So let's talk about this membrane transport. And when we get into later chapters like chapter 10 and 12, we're going to talk about the electrochemical gradient. All right, that actual cells will have a positive and negative charge in certain areas there. And we'll talk about that. All right, so let's talk about membrane transport. And there's two categories when we discuss membrane transport. We have passive processes and active processes, which is basically one involves the use of energy and the other does not. So if you look at this picture here, this could be quite intimidating when you're looking at all these different processes. And I'll tell you, when we're done with this chapter, you're going to know how all these processes work. So we'll see with passive processes, we do not require energy. Active processes require energy. So we're going to start talking about the passive processes, and we'll get into all these different specific ones, all right, throughout the rest of this lecture here, okay? So keep in mind, let's keep it simple. Passive processes, no energy, all right? Active processes, we do require energy. Another quality of passive processes, we're going to move substances down their concentration gradient. What does that mean? Here's our cell. And on one side of the cell, well, that's not even a cell. Don't even worry about that. We're going to divide it in half. Okay? So you have a lot of substance A on our left side of our structure here. And then you have very little of substance A on the right side. So when we talk about a substance moving down its concentration gradient, because nature always likes balance, it wants everything to be equal. So what will happen is substance A will want to move down its concentration gradient. So that means that substance A is going to want to go from the left side of our structure to the right side of our structure. And so it's going to move down its concentration gradient until it's equal, until both sides have the same amount of substance A. That's what we're talking about. That's the concentration gradient. Whereas in active processes, all right, we have to move our substance up the concentration gradient. So that means we're going to go this direction. So that's like rolling a boulder up a hill. So isn't it easier to roll a boulder down a hill? Because once it gets going, it just rolls down the hill. You don't have to push it anymore. Well, think of it like this for the active process. You have to push that boulder up the hill. Guess what that's going to do? That's going to require energy to push that boulder up the hill. And that's what happens when you move against the concentration gradient. So I want to start off by talking about the first type of passive process, and that's called diffusion. All right, so diffusion is the movement of a substance from an area of higher concentration to an area of lesser concentration. I think I have a picture in here. Perfect. Here's a picture. So here you can see we take a medicine dropper that's full of food coloring, and we're going to drop that food coloring into our beaker. Notice that there's a high concentration of food coloring inside the medicine dropper. But when we first drop it into the beaker, in this area here, we have a high concentration of that red food coloring, but we barely have any of it out here. So through the process of diffusion, the food coloring is going to want to move outwards into the areas of less concentration of the food coloring. And you can see it here in this picture in the middle. You can see those molecules are spreading out until we reach what we call equilibrium. Until we reach equilibrium, which everything, all right, will be balanced. I almost forgot about my dentist appointment today. Don't worry, it's not till later. Okay, so that's what we're talking about when we're talking about diffusion. All right, so we're going to have a movement of a substance from an area of high concentration to an area of low concentration. And so that's what we're going to see, all right, when we're dealing with a lot of these ions that we're talking about in molecules. They're going to be moving. And so when we're describing that movement, all right, we describe it as kinetic energy. Kinetic energy is the actual energy in regards to motion and movement. Potential energy is, and the best way I can describe it is, here's a hill. At the top of the hill, you have this big boulder, and it's just sitting there. It's not moving. So that's how we describe potential energy. That boulder has the potential to start to move, all right, once a force is acted upon. Okay, once it's in motion and it starts to roll down the hill, that is known as kinetic energy. Constant motion, all right, or that movement. When something's in motion, then, all right, it's in motion due to kinetic energy. Keep in mind that diffusion will continue as long as there's nothing interfering with it until we reach equilibrium, when all of those molecules have distributed themselves evenly in a given area, like in our beaker here. Okay, so diffusion will keep going until it reaches equilibrium as long as it's not opposed. Okay, so now let's talk about the different types of diffusion. There's simple diffusion. And this is what we're going to see in our plasma membrane when molecules just pass right through the plasma membrane and they're not stopped. Remember what I told you. They have to have two qualities. They have to be very small and non-polar. Okay, very small and non-polar. So they're literally going to move from outside the cell to inside the cell, or vice versa. All right, and they'll go right through the plasma membrane. As long as there is a concentration gradient, that there's a difference, that there's an area of high concentration in one area and an area of low concentration in another area. Okay, so for example, if there's more oxygen outside of your cell than inside the cell, oxygen is going to move from outside the cell to inside the cell, because oxygen is small and non-polar. All right, same thing with carbon dioxide. Okay, so simple diffusion, great test question. It is not regulated by the plasma membrane. Plasma membrane has nothing to do with simple diffusion. All right, the only thing that is going to regulate the diffusion is going to be the existence of a concentration gradient. If there's an area of high concentration and low concentration, then we'll get the movement of those molecules. If there's no concentration gradient, if it's equal on either side of the plasma membrane, we're not going to see any movement occur because it's at equilibrium. It's happy. So that's what we're seeing here in this picture. Okay, you can see that you have an area of high concentration of oxygen outside the cell, low inside the cell. So oxygen wants to move from outside to in. Same thing with carbon dioxide. Lots of carbon dioxide inside the cell, very little outside the cell. So carbon dioxide will leave and go outside. And that's what happens during metabolism. All right, blood brings oxygen to the cells, to the tissues. Those tissues could be exercising, for example, and so they need oxygen for that cellular respiration I was telling you about. So oxygen will move into the cell, and oxygen will go to the mitochondria, and the mitochondria will then make ATP to create energy so it can supply fuel for, like for muscles, for example, those exercising muscles. And as a result of all that, you will start, one of the waste products are going to be carbon dioxide. And so those cells and tissues are going to get rid of the carbon dioxide, all right? And the carbon dioxide will leave the cell, go outside the cell, back into the bloodstream, and then you'll exhale it. Cool stuff. All right, so the other type of passive process for diffusion is what we call facilitated diffusion. In facilitated diffusion, all right, again, we're going to deal with the movements. We need help. We need help. That's why we call it facilitated diffusion in the movement, all right, of molecules across the plasma membrane. In this case, we'll see that these molecules, all right, are going to be small charged molecules or polar molecules or solutes. Okay, so they're small and charged or they're polar solutes. So that means we are going to rely on some of the plasma membrane proteins to help move those molecules across the plasma membrane. And so we have two types: channel-mediated diffusion and carrier-mediated diffusion. Channel-mediated diffusion means we've created these special channels. That's this guy right here. These special channels. And these channels are filled with water because there's water all over the place, inside the cell, outside the cell. And they're going to allow the movement of things across the plasma membrane. Here's the thing. These channels, and you need to memorize this, are specific for one type of ion. Sodium, for example. Okay, so if we have what we call a sodium channel, okay, that means that only sodium can go through. Calcium can't go through. Potassium can't go through. Chloride can't go through. All right, so they are specific for one type of ion. You need to understand that. So when we talk about these special channels, all right, we have a couple different types. We have leak channels, which are always open. Like 7-Eleven, open 24/7. These things don't close. So we call them leak channels because they allow those ions to leak across the plasma membrane, and they'll always go in the direction of where the lower concentration gradient is. For example, you have a sodium leak channel. There's more sodium outside the cell than there is inside the cell. So since these sodium channels are never closed, sodium will move from the area of high concentration to low concentration. It means it goes from outside the cell to inside the cell, and it'll keep doing that. Now, the other type of channel is called a gated channel, and it is normally closed. And the only way we're going to open it is in response to a stimulus. Well, that goes back to chapter 1 when we're talking about our homeostatic model there. All right, you have a stimulus, and then you're going to have a receptor to monitor that stimulus. So we need some sort of stimulus to occur. And as a result of that stimulus, the channel will immediately open. And it's usually only open for a short period of time. So we're going to learn about this process in much more detail when we get into chapter 10 and 12, when we talk about muscle and nerve cells. But for right now, I just want you to know the basics. Leak channels, always open. Gated channels, normally closed. They'll open in response to some sort of stimulus. The other type of diffusion is going to be the carrier-mediated diffusion. Now this, all right, requires what we call a carrier protein. And the carrier protein that resides in the plasma membrane is actually going to change shape once it binds to whatever the substance is that it's allowed to bind to. All right, and when that happens, then it will allow the movement of that substance from one side, the outside of the plasma membrane, to the inside. Again, it's always going to move down the concentration gradient. So when we're dealing with what we call uniporters, that means that these type of transport proteins can only move one substance. Glucose, for example. Okay, we saw that here. Here's what the transport protein does. All right, you have those glucose molecules. All right, they enter into the transport protein, and then it'll literally change shape. All right, so the glucose can enter into the cell. So here you go. I didn't know why I went back. I should have done this. So you can see how it's changing shape. All right, here. And then again here, and then again here. And it changes shape, all right, to facilitate the movement of that molecule across the plasma membrane based on the concentration gradient. So obviously in this example, there's more glucose outside the cell, and so we want to move it inside the cell. Okay, folks, I'm going to stop here. All right, I know it's a lot. I'm going to stop here because I think we've gone pretty far. And if you want to finish up this chapter, okay, feel free to go to my YouTube page, Dr. Kaz, okay, and go under the playlist there, or you can search either under chat sessions or review sessions. And the rest of this material, there's not too much more, all right? But the rest of this material will be present in one of those one of those recordings there. So any questions